Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, The Simple Western Approach to Vaccine and Clinical Protein Research. The birth of the biotechnology industry in the early 1970s provided a growth opportunity for a technique that really came of age during the latter part of that decade. The Western blot is widely used to detect specific proteins in a tissue sample. This analytical method also permits the determination of the molecular weight of a protein. Epitope mapping, spots imprinting analysis, and structured domain analysis are just a few of its applications. But like with all technologies, there is always room for improvement, sometimes much improvement. And that's what our webinar is all about today. We've assembled a scientific panel who will discuss a major advance in the development of a novel Western blot platform. The panel is going to demonstrate how this system addresses the major challenges that researchers face on a daily basis. Things like unreliable data, delayed time to results, reduced productivity, and ultimately more costly research programs. Let's meet our panel. Dr. Peter Fung is the product manager of the Simple Western platform, which is manufactured by the company ProteinSimple. Peter will furnish us with a wealth of details about the Simple Western platform and its range of capabilities. Melissa Ham, who serves as a research biochemist in the area of vaccine analytical development at Merck & Company, is going to tell us, appropriately, about a novel approach to Western analysis in vaccine development. Dr. Alice Fan is an instructor in medicine and oncology at the Stanford University School of Medicine. Alice will discuss the use of the Simple Western in a study involving the application of nanoscale proteomics for profiling tumors, as well as the therapeutic response to cancer. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console, and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So, if everyone's ready, let's get going. Dr. Peter Fung will be our lead-off presenter. Peter, floor is yours.
Thank you very much, John. I'm happy to give a brief introduction to the Simple Western, our gel-free, block-free, hands-free platform from ProteinSimple. In 1979, Tobin, Stalin, and Gordon published this article in PNAS, which described a novel technology in which proteins were separated on a polyacrylamide gel, transferred to a nitrocellulose membrane, and then probed with an antibody specific to their protein of interest, or what's better known now as the Western blot. So while this publication is over 30 years old, the Western blot today is still the most widely used technique for protein detection. But for you, the researchers who have run Western blots, you probably know and understand that Western blots can be very time-consuming and labor-intensive. There's a lot of steps involved to perform from start to finish to complete a Western blot, and often you'll get inconsistent results, or you're trying to get reproducibility in your data can be a challenge with the Western. In the end, the results you get from a Western are semi-quantitative at best. So even though the Western blot is considered the gold standard for protein detection, scientists have had to put up with the limitations that Western blots have, and it's really because they haven't had any other choices or options available for them to use. And that's until now. I'm excited to be able to come to you today to introduce to you our Simple Western product family. This includes Simon, that runs 12 size-based separation assays, Sally, which is able to run up to 96 size-based separation assays, the NanoPro 1000, which is our original platform which performs charge-based separation and can run up to 96 assays, and we recently introduced Peggy, and she's able to run either size or charge-based separation, and she can run up to 96 assays in a single experiment. I should also note that all of our instruments perform separation and immunodetection in a capillary. So with the Simple Western, it delivers for you, the researchers, automation. All the steps, the manual steps involved to perform a Western blot are now fully automated for you. It gives you better reproducibility in your data, and you're able to do, for the first time, true quantitation of your results. This is using our gel-free, blot-free, hands-free Simple Western platform. The Simple Western provides for you new capabilities. Within a single experiment, you're only using microliter sample volumes. You're able to test multiple antibodies or lysates, and you can generate up to 96 data points in a single experiment. What this means for you as a researcher is that you're more able to more easily do your assay optimization within a single experiment, but more importantly, you're able to get more data with the Simple Western. With the Simple Western, so what are the steps involved to perform a Simple Western? Well, first of all, you'll generate your samples as you've done before. You'll denature your material in an SDS sample buffer to generate a lysate. It's pipetted into an assay plate along with a primary antibody, a secondary antibody, and our chemiluminescent substrate, and a couple buffers. The assay plate is placed into the instrument. You hit start and walk away. Within a few hours, you're able to come back to your results automatically analyzed and generated for you. So how does the Simple Western work? I'll just briefly walk through both our size-based separation as well as our charge-based separation. As I mentioned to you, all the steps of both separation and immunodetection take place in the capillary, and all of these manual steps that you would have with a Western blot are now automated fully for you by the instrument. So the instrument will pick up the capillaries, load both our stacking and separation matrices into the capillary. It will then load your sample into the capillary, transfer the capillaries to a separation tray where you'll now separate your proteins based on their molecular weight or size. We then immobilize the proteins to the wall of the capillary using a proprietary UV immobilization capture technology. We can then flush out the separation and stacking matrices from the capillary and perform a standard immunoassay using a primary antibody to your protein of interest, a secondary antibody that's HRP conjugated, and a chemiluminescent substrate. As I mentioned to you, upon completion of your experiment, you'll get your data quantitated for you and summarized for you, as shown by the electrophoregram at the bottom. In the case of our charge-based separation, we don't denature our proteins. We keep them in the native state. We also add the lysate to a sample buffer that contains ampholytes and PI standards. That material is then loaded into the capillary. We then separate the proteins now based on their charge or PI. We then perform the same UV immobilization capture step, locking the proteins in place, and then we perform our immunoassay, and we're able to get our results from the experiment. With the Simple Western, upon completion of your experiment, you'll get three views of your data. The first is our capillary image view. We use a cooled CCD camera to collect the image, so what's shown in the left panel is the actual capillaries and the chemiluminescent signal generated in them. We also generate an electrophoregram, which is the real data that you're getting from your experiment detecting your protein, and we'll have it for either size-based separation or charge-based separation. And finally, we have our lane view image, which is a view that's more similar to what Western blot users are more familiar with. It's a virtual Western blot that represents the electrophoregram data. So just to demonstrate one example or application of our Simple Western technology, what's shown here is pathway profiling. In this particular case, the NF-kappa B pathway was examined using our SALI instrument. What's shown here are 7 different proteins that are involved in the signaling pathway and 1 loading control. What's run in this experiment are 2 different biological conditions, minus and plus TNF-alpha treatment, and we're looking at 2 different lysate samples, a whole cell extract and a nuclear extract, and the entire experiment is run in replicate. So this isn't even running SALI at her full potential, but you can see in trying to characterize signal transduction pathways, it's an easy application to perform on the Simple Western. So for today's webinar, you'll hear how our 2 guest speakers have been utilizing our Simple Western technology for their protein analysis studies. Melissa will talk about her work using Simon to examine vaccine development. And Alice from Stanford University will tell about her experiences using Sally and the NanoPro 1000 for exciting cancer research she's been doing using her precious patient samples. So for us, the Simple Western is the future. It's our gel-free, blot-free, hands-free platform. We have Simon and Sally that do size-based separation, NanoPro 1000 that can do charge-based separation, and we have Peggy that we just introduced that can run either size or charge-based separation, and she can do up to 96 different assays in a single experiment. The choice is really yours. So in conclusion, the Simple Western delivers more. We have size and charge-based separation of your samples. We can do specific target identification as we're doing an immunoassay. For the first time, you can do true quantitation of your results. You can get more reproducible results because all the steps involved, the manual steps involved with performing a Western blot, are now fully automated from you from start to finish. This will provide you with greater time savings and greater throughput, and most importantly, you'll get more data with the Simple Western. With that, I'll say thank you very much for your time and hand it back over to John.
Peter, thank you for that detailed presentation on the Simple Western. You've done a great job in setting the stage for the talks that are about to follow. If you're just now joining our webinar, welcome. As I mentioned earlier, we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Melissa Ham, will now begin her presentation, and it's one that looks at a new approach to Western analysis in vaccine development. Melissa?
Thank you very much, John. So today I'm actually going to give a presentation on what we've been doing to use the Simon instrument to either replace some of the Western blots that we've traditionally run, or as an orthogonal method to some of the other methods that we normally run in our lab. So the outline of my presentation, I will go again just over the assay overview, reemphasizing that everything is done in capillaries. I'll go over general sample preparations, some of the potential applications that we found that are useful for us in our lab, some of the advantages and disadvantages of this technology, some things to consider when developing your methods, and then I will go into individual case studies and then conclude my presentation. So to just reemphasize what Peter had said, the Simon instrument performs separations in individual capillaries. So the first step is that the stacking and separation matrices are loaded into your capillary, followed by your sample. Then voltage is applied to the capillaries and allows your samples to then separate based on their size. Then exposure to UV light causes the proteins to then bind to the capillary wall. The gel matrices are washed out, and then that allows you to perform a normal Western blot by adding your primary and then your labeled secondary antibodies. And then finally, the instrument will quantitate the signal that's present in the capillary from the samples, and can be visualized by either an electrophoregram, which is shown in the slide, or the traditional gel image. General sample preparation. So samples are denatured and reduced in the presence of SDS and DTT, and in our lab we incubate them either at 70 degrees or 95 degrees for 10 minutes. All your samples and reagents are loaded into a 384-well plate. The plate and other reagents are loaded onto the system, and the run starts. The diagram shown below is just a snapshot of basically where everything goes in the plate. And again, one sample is loaded per capillary, so you don't have any cross-contamination issues, because a lot of times when you run gels, sometimes you can get cross-contamination if you overflow a gel, and that's not as big of an issue with this system. Some of the potential applications that we found that are useful in our lab are concentration analysis, antibody screening. We can monitor fermentation and process. It can be used for identity, host cell protein analysis, and many more. And the key is that it can be used as a complementary assay to other techniques like ELISAs. Some of the advantages and disadvantages of the technique. Some of the advantages are it's very quantitative, unlike your traditional Western. It has fast analysis times. We can get samples run in about 3 to 5 hours. The method development is very simple. The sample preparation is very easy. Once you get the hang of loading into a 384-well plate, the plate can be prepared in about an hour, and you need very little volume of samples and reagents to run. Some of the disadvantages are the current gel matrices that are available are not designed for larger proteins, so proteins that are greater than 250 kilodaltons. There are limited availability of secondaries. Right now, only anti-rabbit and anti-mouse are available. And like other Westerns, you need program-specific primary antibodies in order to use this. Some things to consider when developing the methods for this is the primary antibody dilution. Traditional Westerns typically use dilutions of 500-fold or more, and we found that we have to greatly cut the amount of, or really have the antibodies that are less dilute for this technique. So most of our antibodies for Simon are diluted between 20 to 1,000-fold. And one of the issues with some of this is that lower dilutions of the antibodies can lead to higher backgrounds, but you can sometimes provide lower detection limits if you have more antibody present. For your sample preparation, we incubate the samples under the same conditions that are normally used for SDS page, so we typically don't have to do any additional development for that. Our purified proteins can typically be diluted between 0.5 and 20 microgram per mil, and fermentation samples can usually be diluted to between 50 and 500 microgram per mil, or even run undiluted. Some additional considerations. For improved sensitivity, you can actually play around with the sample injection and stacking matrix loading. We found that increasing sample injection times to 12 seconds and stacking matrix loading times to 16 seconds can be quite useful for low abundance proteins to demonstrate clearance. It just gives us an increased level of sensitivity so we can show larger fold clearance. Okay, so now I'm going to go into the individual case studies. So Project 1 is a multivalent vaccine that consists of 4 different proteins ranging in size from about 50 kilodaltons to 300 kilodaltons. Our goals for method development for this particular program is concentration analysis. We ultimately want to be able to determine the concentration of the individual monovalent proteins and then also in the tetravalent final vaccine formulation. It can also be useful for identity and host cell protein clearance. For this program, all the samples were incubated at 95 degrees for 10 minutes. And for our initial studies for this particular program, we just used 2 of the smaller proteins because 2 of the larger proteins were higher than 200 kilodaltons, which is higher than the recommended cutoff. So we initially just focused on the 2 smaller proteins. So this gel image shows what proteins A and B look like. So protein A was expected to be about 48 kilodaltons, and it runs at about 52 kilodaltons. Protein B is expected to be 94 kilodaltons, and it runs at about 106 kilodaltons. And one thing to note is that protein B can be easily clipped, and if it is clipped, it's expected to be around 75 kilodaltons, and it runs at about 86 kilodaltons, and that's something to keep in mind. So one of the first things that we looked at is the linearity of our particular proteins. So protein A is linear over a concentration range of 0.5 to 7 micrograms per mil, and the protein is about 48 kilodaltons. And the image on your right is just the gel image showing the pure standards, and then also 3 samples that we've tested to determine the concentration analysis of. And then we also did the same thing with our protein B sample. So we actually looked at the dynamic range of this sample in particular. So it's linear over about 2 orders of magnitude, or from about 0.2 to about 17 micrograms per mil. So we found that that's a really good dynamic range for us for this particular protein. Next thing we looked at was the capillary-to-capillary repeatability. So in one run, we ran one concentration of the sample in each of 12 capillaries on the Simon instrument. As you can see by the gel image, for some reason in capillary 11, things did not run as planned. And you know, we didn't really think that's too big of an issue because over the 11 capillaries that we got really good peak area CVs of about 6.8%. So ultimately, we want to look at fermentation samples and see how much of our proteins are actually being expressed in our fermentation sample. So if you look at the gel image, on the left-hand side of the gel image is our standard curve of protein B. And then in capillary 6, we ran the clipped version of that protein. And then capillaries 7 through 12 are our samples in duplicate. And as you can actually see, one of our samples is being clipped. We're starting to see that P86 band. And then the next slide is just the electrophoregram images of the gel images on the previous slide. So you can actually see by the electrophoregram image that we can easily see and resolve that P86 band. So we are definitely seeing clipped samples. One of the other techniques that we normally use to monitor these fermentation samples is a reverse phase method. And we use that method right now to measure the concentration of B in these samples. But as shown in the figure, our HPLC method cannot distinguish between the normal protein and the clipped protein. So Simon can actually provide additional information that the current method can't. So I mentioned earlier that this particular project has 4 different proteins in it. And so protein C was the next one we tried to focus on, trying to optimize because it's the largest protein. It's above 300 kilodaltons. So we had made adjustments to the separation, stacking, and injection times to better detect and resolve C. And as you can see by the image and the electrophoregram images, that some improvements have been made, but we're still working out the final conditions. So even if your proteins are heavier, the current matrices that we have, you still can use to detect and resolve those proteins. You just need to make some minor changes and adjustments of things to try to help better resolve those proteins. So we ultimately want to be able to detect and quantitate all 4 of the proteins. So this just shows a mixture of 3 of the proteins where we can, easily resolve and detect. So this can actually be easily multiplexed. We can mix all the antibodies together and detect each of the proteins. We're still working out conditions to separate and detect the trivalent mixture, but this looks very promising as far as detecting all 4 at one time. Another good use for this instrument is antibody screening. So what this image demonstrates here is that we actually tested 10 different antibodies to determine the most appropriate antibody for detecting this protein and using this particular instrument. So as you can see, we can run up to, you can run up to 12. We just chose 10 different antibodies. So we can go and look at this and then choose the antibody in, lane 8 as the best antibody for detecting this particular protein. So another useful method is the host cell protein clearance. Host cell protein clearance needs to be demonstrated during the process to ensure that very little of that protein from the host cells is present in the final vaccine for safety reasons. There's a commercially available monoclonal antibody that exists for this, and also we had a new antibody made against a more relevant capsid protein. And then to enhance the sensitivity of this method, the sample injection was increased to 12 seconds from 6 seconds, and also the stacking matrix and loading was increased to 16 seconds. So this slide just shows the two different antibodies that were used. The one on the left is the commercially available antibody, and the one on the right is the other antibody that we had made. We looked at column feeds, so prior to, so basically the sample prior to being loaded on the column, and then the sample after the column. So we can say that the host cell protein is detected in the sample, but we get about a 3-log clearance up to this step using either antibody. So now I'm going to switch gears and go into another program. So the second project is an OMV-based vaccine that contains at least 5 antigens of interest. Our initial study focused on 2 of the antigens that are 40 kilodaltons and 140 kilodaltons in size. The goals for this method development is purely concentration analysis. These samples were incubated at 70 degrees for 10 minutes. So we looked at the first protein in this particular vaccine, and we wanted to demonstrate linearity to see where it's linear. This particular protein is linear between 1 and 20 micrograms per mil, and its molecular weight is about 39 kilodaltons. And then we did the same thing with the second protein, and we see linearity between 1 and 10 microgram per mil, and the molecular weight of this particular protein is about 140 kilodaltons. And then the next thing we wanted to do is look at the mixture of the proteins. Can we actually multiplex these 2 proteins together? And so we looked at a mixture of the 2 proteins and then tested 3 different strains to see, the concentration of each of the samples. So the different strains express different levels of each protein, but the incubation temperature of 70 degrees was not sufficient to completely reduce and denature S3. So we needed to actually increase the incubation temperature to 95 degrees. And actually, as you can see in the image, the S3 band, a lot of your protein is not really getting fully separated, so it's not fully going in. So that tells us it's not being fully denatured and reduced. And even on the previous slide, testing the one protein itself at higher concentrations, we were also not getting full reduction in denaturation. So this was one case where we actually had to modify the incubation temperature and go against what we normally use for our SDS page analysis. So the key goal for this program is to actually measure the concentration of each of the proteins. So for this particular sample, we had run a concentration range of 0.25 to 5 microgram per mil. All of these samples were tested neat, and then we obtained RSDs over 3 days of less than 2%. The table shows the 2 samples that we tested, the concentration that we obtained by Simon, and also the concentration that was obtained by mass spec. So we had another group run the same samples by mass spec to get somewhat quantitative results, and our results actually match very well with what they were getting by mass spec. So we were quite pleased with that. And then we also did the same thing for the second protein. We measured the concentration of three samples, and the concentration, the standard curve range for this particular protein was 0.5 to 5 microgram per mil. For these samples, because we expected the concentrations to be higher of this protein, we ended up diluting sample 1 200-fold, sample 2 4-fold, and sample 3 is actually run neat. And again, we got really good RSDs over 3 days for those proteins as well. So now I'm going to go into the final case study that I have for you. So our Project 3 is a live virus vaccine consisting of 3 major capsid proteins that are between 60 kilodaltons and 130 kilodaltons. And our goals for this are to detect and quantitate the major capsid proteins in the fermentation samples. These samples are incubated at 95 degrees for 10 minutes. And this is one program where we actually had Western run for this actually prior to running Simon. So this slide just shows a comparison between the Simon results, which are on your left, and the traditional Western results, which are on the right. And what can be said is that the same general trend can be seen in your Simon results and the Western results. So you can look at the RS sample in both the Simon and your traditional Western and say that, you know, the RS material contains more antigen than any of the other samples. So it's, you know, it's a good technique. It basically gives you the same information, but we can actually get that information a lot quicker on the Simon. Since we are looking at multiple antigens for this particular program, we wanted to look at the ability for us to multiplex, to test for all three antigens at the same time. And we expected that all three proteins would be resolved, since Y and Z are expected to be about 12 kilodaltons apart, but we found that they're actually not very well resolved. So it appears based on the gel image that Y and Z are resolved, but if you look closely at the electrophoregram image, they're obviously not very well resolved. So this is why it's important to look at both the gel image and the electrophoregram image before making full assessments. And another thing to note with this is that there are modifications that you can make to try to help improve the resolution, but we just have not tried any of that. So for this, really right now, only the X and the Y or the X and the Z can be multiplexed, but we cannot multiplex all three under these current conditions. So to conclude my presentation, multiplexing can be useful, but it's not applicable in all cases. We were able to demonstrate linearity for all the proteins tested to date, and the quantitative results obtained are in line with other techniques. We can demonstrate clearance of unwanted proteins and monitor them, and we can quantitate them more accurately. Fermentation samples can be easily assessed with minimal sample preparation. And this technique can provide additional information that other methods cannot. And I just want to acknowledge my management at Merck, the bioprocess group, for making all the samples, and Basic Research for providing us specific antibodies. ProteinSimple, I'd like to recognize Chris and Annegret for their discussions on getting these methods up and running, and for Peter and his support. John, back to you.
Melissa, thank you for that incisive overview of this new technology. I think your case studies and list of conclusions is going to be especially valuable for our webinar audience. Before we proceed, let me remind everyone of our Q&A segment that comes right after all the panelists made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Alice Fan, is ready to talk about nanoscale proteomics for profiling tumors and the therapeutic response to cancer. Alice?
Hi, it's a pleasure to be here this morning. I'm an instructor of medicine and oncology at Stanford, and I'm a translational researcher, so it's a real pleasure to tell you today about how I've been using the nanoproteomic technologies of Simple Western to be able to analyze clinical specimens. As a physician, there are several periods of opportunity to make assessments that can aid clinical decision-making. I'm a lymphoma physician, and the standard evaluation of lymphoma at diagnosis involves removal of a surgical specimen and then tissue examination of that specimen under the microscope with histology, immunohistochemistry, and flow cytometry, so we can look at the surface markers to determine what subtype of lymphoma the patient has. And then we look at the overall tumor burden that's present in the patient's body by physical examination and imaging, such as CT scans and PET scans. Now, there's a lot of current research that's really important analyzing molecular signatures in tumors and extremely variable. Most of this focuses on tissue that's taken at the diagnostic time point. So, for example, you have a large surgical specimen of lymphoma or whatever solid tumor, and you're able to analyze DNA by things like karyotyping or all the way into deep sequencing, look at RNA expression and now microRNAs by very sensitive technologies such as quantitative PCR or very high throughput technologies such as microarrays. And then we're able to look at proteins, but this is a little bit where we've been behind. The standard that you've heard about, the Western blot, requires quite a bit of tissue to make a single measurement. Flow cytometry is very powerful, as is mass spectrometry, but it's just really hard to process these tissues in a way to get real-time useful information. And I remind you that changes in RNA, every good scientist knows, must be validated at both the protein and at phosphoprotein levels, because a lot of our targeted therapies now in oncology are inhibiting kinase pathways, and this can only be seen if you look at changes in protein phosphorylation. DNA will never tell you that answer. So we go back to the periods of opportunity. Based on our diagnosis and our diagnostic molecular studies, we make our best decision what to treat a patient with, and then we treat them. It's sometimes a combination of surgery, radiation, and chemotherapy, and then we wait for a clinical response. Right now, when we treat with a chemotherapy, we treat for 3 months, and then the first time we evaluate what's happening is when we reassess the tumor burden by repeat physical exam, CT scan, and or PET scan. And we never really know what happened to those cells. All we know is if the tumor is there or not, or if it shrank or not. So what would be much more important to look at if we had the tools would be to analyze the biologic response. And this would allow us to determine how therapeutic agents actually work in our patients. It's really heartbreaking when a patient says to me, Dr. Fan, how do I know if this is working on my cells? You told me it might work on 20% of patients. How do I know it's working on my cells? And right now, we just don't have an answer better than saying we'll check in 3 months and see if it shrinks. And that's just not good enough. Not good enough for my patients and not good enough for development of novel agents to treat them. So when I say biologic response, I'm talking about profiling changes in cellular pathways of tumor cells that occur after administering a therapy. For example, we would take a pretreatment sample, and this is what we often get, the surgical sample, and we can get a tumor signature. But the piece that's missing is early after starting a treatment, like day 3 or day 5 or day 8, after 1 week of therapy, we would love to be able to re-examine this patient's cells and see what's happening at the biologic level. Are the cells responding in some way? And then correlate that to eventual clinical response in 3 months, where they might now have a normal signature if the tumor is gone. But as we discussed, the barriers to this are the amount of specimen required. To analyze proteins at this level requires quite a bit of tissue. And the processing requirements for things like flow cytometry to get a single-cell suspension is not trivial. And there's a lack of highly quantitative methods. Having something that's very quantitative could help us greatly. And there's an inability to measure complex phosphorylations. So all of these are addressed by the new nanoproteomic technologies that we have now. And I've used the high-throughput, automated Simple Western platform for both size and charge-based separation to profile cancer signaling pathways in patient cells. So I'm going to divide the rest of my talk into 2 parts. Part 1 will be focusing on using the size-based Simple Western and how it enabled me to do rapid profiling of very small amounts of clinical specimen for diagnostic signatures in our patients. And then Part 2 will focus on how I've used the charge-based Simple Western and how that made it possible to determine the specificity of cancer drugs in both preclinical and clinical specimens. So first, using the SALLY platform now, which is size-based separation, I tested the linearity of a protein of interest. This is PRDX6, which is about 25 kD. And loading just nanogram amounts of total protein gave very linear quantification with an R-squared of 0.99. Next, I turn to the very straightforward preclinical studies that we do when testing tyrosine kinase inhibitors. I used HCC 827 cells, which are lung cancer cell lines, and we have a version of it that is very sensitive to gefitinib, a tyrosine kinase inhibitor used clinically, and a version of it that's resistant. And I just looked at the basal signatures of ERK. And what we can see is that if you look at ERK, there are different baseline levels that are significantly different in 3 of the isoforms. So this leads you to hypothesize, okay, perhaps part of the resistance mechanism is that there's a differential response to the tyrosine kinase inhibitors in these 2 different types of cells, the sensitive and resistant. However, if we actually look at phospho-ERK1 using the SALLY platform, we could see that both types of cells still were able to decrease phospho-ERK1 when you treated with gefitinib. So this is a really important example of where looking at just a single time point might lead you in one direction, but then it doesn't necessarily follow that the hypothesis of inhibiting that pathway will be the same. So then the next slide shows that when we looked at other pathways, in this case we looked at both ERK and then the AKT pathway, GSK3B, we can see now, looking at the GSK3B, there's actually a decrease in cells that were sensitive, but it didn't significantly decrease. In fact, it increased in cells that were resistant. So this hints more that there's an inability to inactivate the PI3 kinase and AKT arm of the pathway. And so this is the approach I would like to take in my patients, analyzing both the baseline time point, but also seeing how they change over time, because that will give us many more insights on how resistance is actually happening. And so I'm going to turn my attention to renal cell carcinoma, where there are very similar pathways where you have a receptor tyrosine kinase, and you can see on this slide that there are many different both receptor inhibitors as well as small molecule inhibitors downstream of these pathways. And all of the drugs listed here are actually used in kidney cancer, renal cell carcinoma. And so you can see it would be very useful to be able to quickly and in real time profile these molecules in patients with renal cell carcinoma. And this is an example now using clinical material on Sally. I was able to compare both frozen tissues from the tissue bank from patients with RCC to fine needle aspirates from patients with RCC. And much to my delight, I could use Sally to make multiple measurements overnight in one FNA specimen. Generally, if I had one FNA, I'd be able to run one lane on one Western and make maybe one or two measurements if I stripped and reprobed. Here, because we can use a 96 sample format, we're able to run one sample and do multiple cycles and probe with multiple antibodies. So you can see I was able to very rapidly profile AKT, both 1, 2, and downstream GSK3 beta and S6, as well as ERK and PRDX. And I was very happy that I could analyze both fine needle aspirates and frozen tissues. So then I really tested it, and I got a lot of samples out, and I tested 20 FNAs. And I profiled up and down the MAP kinase, PI3 kinase, and STAT pathways. I was able to analyze all 20 of these specimens in 2 overnight runs, which basically means about 1 hour of setup per overnight run. Then you put it unattended into the instrument for 14 hours, and then in the morning you come back and you have all this data. So now it's really possible to have a next day profile of patient specimens. And this is a really striking result. So when I looked across 200 specimens across many different tumors now, we can see that baseline ERK1 expression varies quite widely. And this really gets at the difficulty in trying to develop biomarkers that can predict how people will do based on a single time point prior to diagnosis, because the set points for tumors are all very different. And every individual tumor might have a different threshold, but that doesn't mean you can't inhibit the pathway with a kinase inhibitor. So it's very hard to predict, and I think this is where some of our proteomic biomarker tests right now are really having trouble, is to set a single threshold above which they'll respond and below which they won't respond is difficult simply due to the biology of the different tumors. So size-based separation is making it possible to profile proteins in real time in large panels of patient specimens. And now the next step is to be able to analyze FNAs that are taken serially before and early during treatment to see how they change with treatment. So there's a lot of promise for the platform of Sally. And now I'm going to turn to charge-based simple Western and how it is now possible to determine the specificity of cancer drugs in preclinical and clinical specimens. And I would just say at this point that I've had much longer experience with NanoPro 1000 since it came out first. And so charge-based, I am able to now show you the data of these serial measurements. So I trained in the Felshire laboratory where we used very elegant transgenic mouse models to help develop and test new potential therapies and diagnostics. I'm going to tell you about 3 examples right now. One is atorvastatin, which is Lipitor, a cholesterol-lowering drug, and we studied its anti-lymphoma properties in our transgenic model of mice, and I will tell you how I designed a clinical trial based on these results. Another one was using an old drug, doxorubicin. It's been around for decades, and we use it for lymphoma, but trying to deliver it in a new way. We conjugated it to carbon nanotubes in collaboration with the chemistry lab and found that we could deliver it better. So the mouse models have been very effective in testing old drugs for new purposes, old drugs that are being delivered in new ways, and third, new drugs. So I'm also going to tell you about a bryostatin analog that we tested for the first time in mice. This was a drug that was developed by the Wender Laboratory, chemistry laboratory at Stanford, and it has very promising preclinical results. In addition, I'm going to tell you about how important it's been to have the ability to measure biologic response in developing and testing new potential therapies and diagnostics. To be able to measure what happens once you start a new drug and what it does to the individual cells and how that can correspond to clinical response, you need to be able to make these measurements in a very sensitive way over time. So as I mentioned, quantitative PCR to measure RNA is kind of way ahead. We're very good at measuring changes in RNA in a very sensitive way. And I was able to help develop a new way to measure microRNA in just a drop of blood. But really I'm focusing here on proteins. So I'm going to tell you about using nanoscale proteomics in a clinical setting in a phase 2 study of non-Hodgkin's lymphoma of atorvastatin. And also, one of our newest papers is using nanoscale proteomics in myelodysplastic syndrome in a phase 2 study of a novel kinase inhibitor in that setting. So just to remind you, we're now talking about charge-based separation. So if you've seen any of my publications, I call this NIA, the nanoimmunoassay. And so when you see the words NIA in my papers, I'm referring to NanoPro 1000 and charge-based separation. And just to remind you, using a single antibody that's able to measure total protein, because it's a charge-based separation, we can now resolve even a single phosphorylation difference based on the way it's separated in the capillary. So now, for example, I show you ERK1 and ERK2. These can be measured with a single antibody that can detect total antibody, total ERK1, 2, and measure all of the parameters listed on the right. You can quantify single phospho-ERK2, double phospho-ERK2, as well as the relative ratios of unphosphorylated to phosphorylated. So the first thing I wanted to do most recently was to see how sensitive this could really be. And the charge-based separation is extremely sensitive. Adding now a tertiary step of an additional biotin streptavidin amplification allowed us to start with 100 cells in our Eppendorf and get very clean signals this is for ERK1, 2. And it was also very important because I'm going to be analyzing clinical specimens to understand how much lead time you have between collection of the specimen from the patient and bringing it to the lab for processing and batch analysis. And so previously, I had tested doing fine needle aspirates, putting them on ice, and we could transport them for up to 1 hour on ice without having any degradation of signal. But if we're thinking now of doing clinical trials that are perhaps at multiple institutions, we need to do better than that. So I most recently tested the stability of bone marrow, and what we found is that bone marrow cells can be stored overnight on ice prior to isolating the rare CD34 cells in the marrow and then doing a charge-based nanoproteomic analysis. So you could see that in the blue, when we did an immediate separation straight out of the patient, we could measure very clean signals of AKT2 and ERK1, 2. And then we stored them overnight on ice, so that allows for overnight shipping on ice. We could then separate the CD34 cells once they arrived at Stanford and, again, get very good signals. So this is, in fact, the protocol we'll be using for a phase 3 study that's going on internationally. So now to get to some of the data. We showed in mice that statins induce lymphoma regression and apoptosis in our preclinical transgenic model, and this is due to inhibition of ERK, AKT, and STAT3/5 phosphorylation. Here you can see the bright green tunnel staining after 8 days of atorvastatin showed a very brisk apoptosis in response. So I designed a phase 2 study of atorvastatin, the same drug, in patients with low-grade or refractory non-Hodgkin's lymphoma, and I built into the study taking both a preclinical sample pre-therapy, day 1, and then starting 80 milligrams of atorvastatin daily, and then day 8 the patient comes back and gets another sample. Because we can get away with doing a fine needle aspirate, they don't have to undergo a surgery or anything. We just stick a needle into the tumor at the bedside, take out cells, and analyze them. And the goal is to determine if atorvastatin does indeed inhibit the RAS pathway in tumors, which is what we had seen in mice, and then to analyze downstream phosphoproteins of AKT, ERK, STAT, and NIC. So I'll just show you one piece of data that we published in 2009 showing that NIA analysis uncovered decreases in oncogenic signaling in patients that were treated with atorvastatin. You can see that day 8, both phospho-STAT5 and STAT3 dramatically decreased. Now, very interestingly, we were able to use flow cytometry as an orthogonal method to determine what was happening in both the tumor cells and the non-tumor cells. So when we stained for CD20 positive cells, which are the tumor cells, you can see that both phosphostat3 and 5 decreased as indicated by blue, but I'll point out to you that I had to crank up the scale. It's on from -1-fold to increase to 1 + 1-fold. So it's a very subtle difference that we can barely detect by flow cytometry. When we gate on the CD20 negative cells, so these are the non-tumor cells in the sample, we can see that there was an activation of the pathway. So now we can start actually studying what's happening in tumor cells as well as the immune response in the host. And another example from this study is where the nanoproteomics are simply much more sensitive to determine the degree of inhibition. So if we did flow cytometry looking for phospho-ERK using an antibody that recognizes phosphorylated ERK, we can see that there's barely any difference between the untreated and the treated when normalized to control. However, when we do the charge-based separation, we can see that every isoform had a significant decrease. So then I'm going to show you another clinical example of the suggestion that we can now measure percent changes in patients over time. So imatinib, we know, reduces phosphorylation of ERK. And when we did the charge-based separation on cells from a patient, we were very surprised to find that all of the phosphorylated ERK was in a monophosphorylated form. It was monophospho-ERK2. And this is what changed when the patient was treated with imatinib. You can see the reduction of that one peak. When we looked across a panel of specimens, we noticed that all the patients that ultimately responded and had a clinical response were all able to inhibit that single peak. The percent monophospho-ERK2 decreased. In the patients that were relapsed or refractory, they were not able to have a decrease in that monophospho-ERK2. So this gets at several points. First, if you look at all of the untreated time points across the panel, you see once again there's a very large difference in basal expression. So it would have been very hard to set a single threshold above which and below which we could predict patients would respond. It was really the change over time that was found to be significant. So are there specific phosphoisoforms that can be preferred in other therapeutics? I've given you two examples, atorvastatin and imatinib. But when we want to develop novel targets and therapeutics, we would like to develop potential biomarkers in both preclinical and early clinical trials. So is there a drug that we can test in the preclinical setting? Well, I mentioned bryostatin analog, Picolog. So bryostatin is an anti-lymphoma drug that was being studied, but it's a natural product, and it takes 4 tons of sea sponge to generate 1 gram of clinical drug. So you can imagine that it's going to be very impossible to harvest enough drug to test it in full clinical trials. So even though it showed promising activity in lymphoma, the clinical trials were stopped because they realized they'd never have enough drug to test. So the Wonder Laboratory at Stanford had just been very passionately pursuing ways to make analogs to bryostatin in a much more straightforward way. And their lead compound they've called Picolog. They're eager to test it in the first-in-mouse studies. So in collaboration with the Felcher Lab, we tested this compound, and we did indeed see that this compound was very, very potent, and it could inhibit lymphoma growth. We wanted to validate the mechanism by which it was working, and we know that bryostatin is known to activate protein kinase C, and downstream protein kinase C is MEK1. So we used the nanoproteomic assay, charge-based, to see what happens when we treated our cells with Picolog. And in fact, we did see induction of phospho-MEK1. And what's really interesting is, once again, we get a hint that there can be preferential induction now of specific isoforms. You can see that it's all in the right-sided phospho peak. That's where all the induction was. So we're getting, even in the preclinical setting, additional examples whereby charge-based separation can give us evidence of very, very specific drug activity that we couldn't have predicted with any other technology. And I'll give you one final example, which is a novel kinase inhibitor, regurgitab, also known as Onconova 1910, which is a novel multi-kinase inhibitor and is being studied for myelodysplastic syndrome. At Stanford, led by Peter Greenberg, there's a phase 2 clinical trial that enrolled 13 high-risk MDS patients, and they were given this new drug. 1 patient had a partial response, 8 had stable disease, and 5 progressed. And in this population, the median overall survival is usually very, very low, so extending it up to 10 months was actually pretty amazing. And it shows us how much more work we have to do in this field. When we looked at CD34 cells isolated from the marrow of these patients before and during treatment, we could see, this is an example of 1 patient, when we looked at AKT2, which is one of the kinases that are inhibited, we could see that CD34 cells showed a decrease in several specific peaks of AKT2. These are phosphorylated peaks. We look on the lower right, you can see that a lot of the decrease was due to peak 4 and peak 3. Well, when we looked across the panel of patients that we had access to, what we found was pretty interesting. All the patients that had stable disease as a marrow response had a decrease in phospho-AKT2, and those that had progressive disease had stable or increases in that isoform. And if we narrow it down even more and look at the different isoforms, all of that could be attributed to isoform 3. So now we are going to validate this result in a multi-center phase 3 study, and that's why I had done the CD34 condition studies. So in summary, nanoscale immunodetection of proteins, both size-based and charge-based separation technologies, are enabling highly quantitative profiling of patient tumor cells before and during treatment. Profiles of individual protein isoforms and their relative abundance can be used to interrogate the degree of biologic response. I gave you 3 clinical examples. Atorvastatin in lymphoma, we saw evidence of decreased STAT signaling. Imatinib and dasatinib in patients with chronic myelogenous leukemia, we saw the decreased monophospho-ERK2, and that was associated with the clinical response. And then lastly, I just told you about regurgitab in myelodysplastic syndrome, where decreased PI3 kinase was associated with stable disease. Basically, in my preclinical work, these technologies are making my life a lot easier. It's automated, it's sensitive, it is very precise. But in my clinical translational work, these technologies are actually making my research possible. I could not have analyzed these very precious small clinical specimens prior to this. So in conclusion, nanoscale analyses are helping with diagnostics and therapeutic monitoring for both hematologic and solid tumors. And I focused a lot on the diagnostic time point, novel targets and therapeutics, and biologic response, but you can easily envision how this could also be used to look at very small amounts of tissue from minimal residual disease states or very rare populations like cancer stem cells. And even in the field of diagnostics, where we can now start analyzing smaller and smaller bits of tissue, this can really help push back early detection of tumors. So I wanted to thank everyone that has been involved in this work. It's been a lot of different teams from both Stanford and the Felsher Laboratory and the Cancer Research Institute, as well as immunology, chemistry, statistics, and cooperating institutions such as Onconova with the regurgitab study, and of course ProteinSimple. In particular, I'd like to thank Anna Gret, Jessica, Deborah Brita, and David, who have always helped me with my scientific questions, as well as Peter and Jocelyn, who have helped me with all of my other questions. Thank you very much. And now, John, back to you.
Alice, thank you so much for that fascinating presentation. And thank you, Peter and Melissa and Alice, for all 3 of you not only opening a technological window, but for letting us look inside to see how this new, simple Western platform actually works. And it looks like you all hit a resonant chord with our audience. We've received a number of great questions. So let's get started with our Q&A segment.
Okay, Peter, we have the first question for you. Are capillaries replaced frequently? And how do you get rid of cross-contamination issues in capillaries?
So thank you, John. So in our experiments, the capillaries are actually a single-time use reagent or consumable here. So it's really one experiment is run in the capillary, and then it's disposed of.
And we have another question, Peter. Do you mix the protein sample and antibodies in a single tube and then load?
Yeah, so for our assay setup, what you'll do is each of the different components, the lysate, the primary antibody, the secondary antibody, the substrate, and our separation matrices, all go into individual tubes in the 384 wells of the 384 well assay plate. So they'll each get their own row within the assay plate.
Thank you very much, Peter. And Melissa, why do you incubate your samples at 95 degrees centigrade sometimes and at 70 centigrade at other times?
So our incubation temperatures are actually based on the data that we got from SDS Page. So our group spends a lot of time trying to figure out the best incubation temperature and time for those particular assays. So we basically just took their data and adapted it, to this particular assay. And there was really only one case where we needed to change from what we normally use for SDS Page to go to a higher incubation temperature.
And Melissa, what are the program-specific antibodies needed?
So the program-specific antibodies are the ones you need to be able to, to detect your particular protein of interest. So just like a Western, you need to have, the antibody that you normally use for the Western can then be used for the Simon instrument so that you can actually detect your specific proteins.
And Melissa, are all the samples in the 386 well plate run simultaneously?
So on the Simon instrument, you can only run up to 12 samples, so you really use a small portion of that 384 well plate. And yes, everything is run simultaneously, so each sample goes into its individual capillary and it's run at the same time on the instrument.
And Melissa, how about incubation washing steps?
Everything is done actually on the instrument, so the instrument will run through its program. So after your sample is placed in the capillaries, everything is done automatically. The incubation and the washing steps, you can actually change some of the parameters to shorten them, but everything is done automatically on the instrument.
And Melissa, was that purified protein that was utilized for the standard curve, which generated an R2 of 0.99, or was it an experimental sample?
Everything was actually purified proteins. So for one of the programs, we've actually had proteins made specifically to be used as reference standards.
Okay. We have another question, Melissa. Just let me see this. Hold on. Actually, we're moving over to Alice. Alice, same question for Melissa. Was that purified protein that was utilized for the standard curve, or was it an experimental sample?
Yeah, so exactly for my data, when I showed the PRDX6 protein that was 25 kD and got the R squared of 0.99, that was actually using a cell line, so it was an experimental sample. I did not use recombinant for that particular experiment.
And Alice, here's a long one. When you normalized the protein values across the capillaries to tubulin, did you probe with antitubulin in the same capillary that you also probe for ERK, or did you run parallel capillaries for the ERK and tubulin?
That's a great question, whether I normalized using tubulin in the same capillary or in parallel capillaries. And the answer is in parallel capillaries. Long question, short answer.
And Alice, another one. Do you have a list of validated antibodies that you have worked with this equipment?
Sure, that's a great question about validated antibodies. So actually I should say first that ProteinSimple has a list of all their validated antibodies in very nice assay sheets available on the web at their website, and so that's always a starting point. And then for myself, I then start with their assays that have been well developed and then see whether they need any tweaking for the clinical specimen use. And I'll turn it over to Peter if he wanted to add any additional info.
So I'll just add to that at ProteinSimple we have a growing list of different targets that have been detected. So either consult the website, as Alice just mentioned, or as well contact us directly, and we can help guide you on different targets detected with our Simple Western platform.
And back to you, Alice. How do you normalize protein load for the patient samples, or is it only the ratio of the isoform phosphopeaks that you're interested in?
That's a great question, because when you use the charge-based separation, you can get both types of information, and I routinely do both. So when I want a normalized value, I will normalize either to something like tubulin, actin, or HSP. And what's really unique, though, about this type of experiment is that you can get relative ratios. So in that case, you don't need to normalize, and you can simply calculate the relative differences between peaks and different isoforms within one capillary without normalization. So I routinely do both types.
And Alice, what instrument was your clinical treatment assessment done on?
So the 3 clinical trials that I've published and that I presented today were all done on the NanoPro 1000. And the data that I presented at ASCO and ACR, looking at different FNAs from solid tumors, had been done on the SALI.
And Alice, another little bit of a lengthy question. How are the identities of the mono, di, etc. phosphorylated proteins positively identified by antibody recognition and PI alone in the absence of size mass confirmation? How does one weed out false positives?
That's a great question about false positives. So validating what your peaks are is very important. So there are many different things. You can activate some of the proteins and stimulate increased phosphorylation and see that the peaks get higher. Also very important, we treat with phosphatase and see that the phosphorylated peaks go down. And also when there are different antibodies available, like phosphospecific, we also probe with those. And we are just now getting to some very exciting experiments where we can directly validate these peaks using mass spec. So more to come on that.
Thank you, Alice. Peter, back to you. What is a turnaround time from start to finish?
Yeah, so in the case of Simon, it's about 3 to 5 hours to complete your experiment. And with Sally and Peggy, and the run time from start to finish is about a little under 19 hours.
And Peter, another question. With the Peggy instrument, which is capable of either size-based or charge-based separation, can both kinds of separations take place simultaneously within one run?
Yeah, so with Peggy, our latest addition to the Simple Western platform is she has a choice to either run size or charge, but they are in separate experiments, so they aren't run simultaneously in the same experiment.
Well, thank you very much. Unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for the outstanding presentations, and I want to give a big thank you to our audience for your attention and for your extremely thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, ProteinSimple, who made this event possible. Shortly, we'll be sending you a survey, and we would appreciate your feedback on this webinar. Please look out for it, and kindly give us your thoughts, as this will help us continue to bring you topical and timely webinars in the future. Thank you for attending.